Okay, thanks a lot. So, what I will talk about today is drug addiction and more into what happens in the brain when we use drugs and become addicted. And the first question one might ask is, of course, why study addiction at all? And the first answer to that would be that it's a big problem for society. And I think this is clear to anyone, but it's maybe easy to underestimate. I mean, of course, you have the addicts that are whose life is completely ruined by the drug. You can see homeless people or just people drinking a bit too much so that they cannot have a normal job or not a normal relationship. This is quite a lot of people, but it's not an enormous amount, of course. But on top of that, you should also add all the health problems, death and disease that is due to nicotine use. If there would be no addiction, there would be no smoking, I think. You should also add quite a lot of the alcohol-related diseases and deaths. And now we're rapidly scaling up a lot. I don't have any numbers here, but I think you can imagine pretty well. On top of that, we should actually also add a lot of accidents and a lot of crime when people try to get drugs, or that when people are high on drugs. And we should also add the suffering of relatives to all the addicts. If you have a daughter who is on the street on drugs, that's not a nice situation. You would probably prefer a broken arm or a bit of atherosclerosis. So I mean, if we add all this up, it's quite obvious that addiction is a very bad thing, and there is a lot of ways to try to handle that. But I think that one important part of it is to actually understand what happens in the brain of people that become addicted, because then we might have the possibility to stop that in the long run. And we are not there at all, but what I want to do today is to kind of show a bit what do we know about what happens in the brain when people take drugs. And if we start on the very basic side, what happens when you take a drug? It's apparent that what we do is that we play around with the brain chemistry. And neurons, they talk to each other with chemical signaling molecules. And when we do this, we kind of shake the brain in the very foundations. And we could, in principle, do that in a lot of different ways. But if you put chemicals to the brain that just make you feel sick or makes you vomit or have involuntary movements, you will most likely not do that again. What's special with addictive drugs is that they give effects that you like in the short run. So how can they do that? Well, what we know that is that all addictive drugs activate the dopaminergic system in the brain, or rather mimic the activation of the dopaminergic system in the brain. This is so-called reward system of the brain. So here you see a human brain. In red you have dopaminergic neurons. And in particular we are now talking about this cell group in the ventral tegmental area projecting to the nucleus accumbens in the forebrain. This is an extremely important part of the human reward system, and also in all mammals, I would say. If we go a bit more into the details, we can say that all addictive drugs, they cause an increased concentration of dopamine in the nucleus accumbens. So here. So in that way, the rest of the brain, you can say, perceive this as an activation of the dopaminergic cells. So how do the brain interpret this, or what is the result? Well, the result is wanting. That things that increase the levels of dopamine will induce wanting, that is, in the future you want more of it. And what is this system good for? Well, it's important for us to survive, or was at least, still is, but it's even more obvious if you look at it in the evolutionary perspective. So what is important for us to survive? Well, of course, it's food, it's drink, beverages. It's having sex so that there will be more of us in the future. And it's basically a lot of other things. What is very important for humans is apparently social interaction also on top of this. So if kind of people tell you that you do something well, that's a very strong reward for humans. Money is also a very strong reward for humans. But back in the days we were walking on the savannah in Africa, this was not around. So the natural rewards are maybe more easy to think of in this perspective. If we would not go for this reward, we would not survive. It's also important here to put in a learning aspect. It's not only important that we like food. We should also be able to kind of, if we get food at a certain place, if we find a good banana tree or whatever, it's important for our brain to keep track on that. Where can I find it next time? And if you see the tree from a distance, okay, then you should make the link. This is the tree. This is a good place. I should go for it. And this is basically the circuits that the drugs will hijack. So that's what makes them drugs so highly motivating. On top of this, one should also mention that the dopaminergic system is pretty sophisticated in one sense. That is, in the beginning, activation of the dopamine system will kind of tell you that you just had something rewarding. After a while, this will shift to reward prediction if you repeat the same reward. So if you expect a decent meal, for example, and then you have a decent meal, the dopaminergic cells will not fire that much. But if you have an extremely good meal, when you only predicted a decent meal, which can happen in restaurants, then your dopamine cells will fire because you reached a reward level that was above what was predicted. This now becomes a problem when we think about the drugs, because what they do, as we said here, they all increase the concentration of dopamine in the nucleus accumbens. That is, even if you expect the drug to be good, if you just think of it in a kind of computational way here, it will hijack the system and it will elevate the concentration of dopamine, whatever, even if you predicted it to be good, which the kind of signal that the rest of the brain takes home is that, oh, the drug was even better than I thought, although I thought it was good. And this, one thing, is one reason why the motivation to the drugs gets so enormous after a while. So how can they do this? If one would just look at the drugs, you take cocaine, morphine, alcohol, it's all very different kind of things. How can it be that the results of them are so similar and that they activate the reward system? Well, if we start to look at where it all begins, we pour them into our body in different ways, and then they bind to molecules in the brain. These molecules that they bind to are very different. So, for example, we have the opiates, like morphine, heroin. They will bind to opioid receptors of different kinds that are normally bound by endogenous opioids that act as transmitter molecules in the brain. Then we have the psychostimulants like cocaine, amphetamine. This acts directly on the dopamine system by blocking the dopamine transporter or another transporter for monoamines. We have the nicotine in cigarettes and such kind of things that will bind the acetylcholic receptors. We have alcohol that acts on a lot of things in the brain, but mostly glutamate and GABA neurotransmitters. And we have the cannabinoids that bind to endogenous cannabinoid receptors in the brain. So now we have a bit of a puzzle there, right? We have a lot of molecules binding to completely different things, and they all elevate dopamine in the nucleus accumbens. How can that be? Well, they do that in a lot of different ways. And we should remember that when I present it in this way, it's of course actually the other way around, that these drugs became addictive because they had the feature that they activate the dopaminergic cells. So it's in a way not so strange that you pick up a lot of strange classes that do it in different ways, although it might seem very strange when you first see it. So Can start with the easy ones, cocaine, amphetamine, and so on. Here we would have the dopamine cell in the midbrain, the ventral tegmental area. Here is the nucleus accumbens in the forebrain. Cocaine and amphetamine will block different transporters here, and this will lead to an increase in dopamine here, since it's either not taken back or it's leaking out. So the result is increased dopamine. When we come to, for example, morphine, it actually acts mostly down here. This is a neuron that is GABAergic, so that is an inhibitory neuron. So this neuron inhibits the dopaminergic cell. So it acts like a brake on this cell. It acts like a brake on the reward system. What morphine does here is that it binds to a receptor that has a negative action on this cell. So it removes the brake on the dopaminergic cell. So this means that the reward system is more easily activated. The same holds for benzodiazepines. It also acts here in an inhibitory way. And other drugs also act in either here or through afferent coming in here, so terminals coming from other neurons. But in the end, the net result is increased dopamine here, either through inhibition there, activation there, or a direct action up here. Now one could say that this system is, from the beginning, a kind of a nice system, a reward system. We should not fight our reward system. If the drug can activate the cells so that we feel really well, we get euphoric, or that we really feel that we get the things we want, because this is also, as I mentioned before, a kind of a wanting system. Then maybe we should be happy, because life is in a way a lot about doing the things you want and trying to feel as well as you can. However, the activation by drugs of this system has turned out to be not very good in the long run. So here I found on the net was some catchphrase here I liked. A short-term good feeling in exchange for the steady meltdown of one's life. So how can that occur? We activate a reward system that is a good system in a sense. Why does it turn out to be the meltdown of our life? First, we should say that this is not really true for everyone. I guess most people nowadays will occasionally drink alcohol or try to smoke once or twice. And most of us are not addicts. So. But if we look at the processes that take you to addiction, we can say that drug take usually starts with recreational use. That would be you use it now and then, maybe on a party or so. Some people will continue down to making this a habit, so take it every week or whatever, or every day. And after a while, you might come into a compulsive use where you do not have control over the intake anymore. Usually people who reach this stage will also have periods of abstinence, so being off drug. But unfortunately, in a lot of cases, there will be a relapse, so that they will basically come back into drug intake again. Now, what one should consider here when talking about drug addiction is that this risk of relapse is more or less lifelong. It's of course highest in the beginning, but it's a lifelong risk. And also, when relapsing, most of the addicts who were here will kind of directly come there again. It will not be recreational again. And what this shows, in a sense, is that there is something very permanent in the brain of addicts that stays over years and leads right back here. And this is something that we would like to get rid of if we could. Another thing that is important about relapse is that they are often triggered by something. And this can be things like a cue. And a cue is something that was associated to drug intake. So this can be a friend that you took drugs with. It can be a place. So if you go to a bar where you used to drink, it can also be a small dose of the drug, so that you would have a glass of wine at the wedding or whatever, and then start again. Stress is also a major risk factor for triggering relapse. And yeah, drugs, I come back to, it's a kind of a cue, but formally not a cue, of course. As mentioned a bit before, a lot of people stay in all these stages. It's not too easy to know why some people will progress and some people will not. We know that it's a lot of genetic factors involved. People usually say that about 50% of addiction is genetic, 50% not. I guess that is not entirely easy to kind of know, but somewhere around it. Here we can see another plot on how this kind of progress into addiction, or regress, I would rather say maybe, putting in some more factors into it. So we go from use to heavy use, to an early dependence, to a late dependence. So this would be a more true addiction. Starting out, we start with wanting the drug in a kind of positive way. So there is a craving for rewards. Then people take the drug and like it. And after that, they are kind of neutral again. And then next week or whatever, they again feel that maybe it would be nice with a glass of alcohol, or 2 or 3 when it comes to intoxication, maybe quite much more. But it's still nice. Now over time, these things can change a bit, so that the basal state of the addict is negative. So that you would start with a negative effect, and you would use the drug rather to get a relief from the bad situation. And you manage with that, but you come back into this negative side. So here you move from a kind of impulsive stage with reward craving to a compulsive stage with relief craving. And here you have lost the control, and you have also entered to this very negative state. Now, drug addiction is a complex thing, and to look at it from a neurologic or a neuroscience perspective, I'm a neuroscientist, not really a clinician, but I think one can dissociate it to some extent into 3 major parts. There are most likely more of them, but I think these 3 are kind of important. First, there is one part that relates to pathological motivation. That is, you want the drug much more than is making sense. This is on expense of other things. This is a typical thing you can see in addicts, that the wanting of the drug has kind of pushed out other things out of the life. If we look in the brain, it seems very likely that this aspect is in some way dependent on dysfunction of the mesolimbic dopaminergic system. So that was the reward pathway I was talking about earlier. And their targets. I mean, we know for sure that the beginning of addiction is triggered by activation of the reward system. We don't know if that's how you come into it. Or I should not say the beginning of addiction. In one sense, yes, and in one sense, no. You come into it in that way because that's what makes drugs addictive. Then a lot of other things will happen in the brain, and it's not clear that all of that is happening in the reward system. It can be also other things. So therefore, we have the mesolimbic dopaminergic system and its targets here. Another important aspect is loss of control. Because if you have a pathological motivation, you could argue that maybe you can still keep that under control. And it seems like, first of all, this is blown up, or this is expanded a lot in the addicts, but also the module for keeping control seems to be weaker after some time of drug use. And in some cases, of course, if your control module is already a bit weak from the start, of course, then there is a higher risk that you move into addiction. And if we look at the structures here, the prefrontal cortex seems to be especially important. In particular, or it has been shown that in addicts there is a reduced activity in this area. So we have the motivation, we have a control module that is not really able to fight this motivation, and then we have what I was a bit into before here, what is called the dark side of addiction. And what that means is basically that after a while of drug use, the addict will come into this very a state of stress and depression that you want relief from. And if we should look at the neuroanatomy there, the amygdala is probably a key culprit. The amygdala is a place that is very much into fear. Everything from this kind of innate fears from predator birds and snakes in some animals to the fear that we experience with when giving a lecture or whatever, in a sense. So here we have already quite a lot of anatomical structures and different concepts about addiction. And we believe today that these all kind of collaborate to drive addicts into compulsion. I would guess that the balance between these would be a bit different between different kinds of drugs and different people also. I will continue along the motivation track here and basically leave these two. They are also important, but I will not stick to them now. If we look at the motivation, a very influential line of thinking has been that reward learning is involved in drug addiction. And here you would say that long-term associative memory processes may be at the core of addiction. This relates to what I talked about before, about here we have an apple tree and not a banana plant, but it's the same. The brain is very important for survival, to keep track of things that gives you survival value. Either it's a tree or it's a place where you can find mating partners or whatever. The idea that drugs would hijack such mechanisms fits very well with a lifelong risk of relapse, because we all know that memories, they can actually stay on for a very long time. It does not fit with a very simple idea that I think was original, that the drugs go into the body and then they kind of change the homeostasis so that the body now needs the drug and you have to refill it all the time. Then you would just take a month off drug and then everything would be solved. This could never explain lifelong risk of relapse. But this model can. It also very well predicts that cues would be important. That is, things that you associate to the drugs, that they could make you go back on drugs. Makes a lot of sense in this context. And if we look at the anatomy there, what has been, where most of the science has been done is here in the nucleus accumbens and the striatum, which is, or the nucleus accumbens is a part of the striatum, with dopamine coming in from the ventral tegmental area and glutamate, which is the bread and butter, the transmitter of the brain, coming in from the cortex and a lot of other places. So we can expect these things to happen here and here. So how could they look? If we now look at addiction and reward learning, what kind of molecular changes can happen that could explain addiction? Well, we could have changes in gene expression. We could also have changes in the strength by which neurons are connecting to each other, that we call synaptic plasticity. We could also have structural changes. We could have new pathways made or old pathways going away, dendrites of the neurons being taken away, and so on. So I will now move into one of these aspects which I have studied most, and that's synaptic plasticity. It's of course related to the other ones because synapses are made by gene products and they are very close to being structures. And I will talk a bit on what we know now about the cocaine-induced plasticity in this system, the dopaminergic cells and their target. So the approach I have used in my research is mouse-based. This is basically because we can do a lot of things in the mouse. We can take away certain receptors or signaling molecules in specific places in the brain or in specific types of neurons, one can say. As a stimulus, I've looked at cocaine. We know that the mechanisms that behind addiction is not identical between the drugs, but we assume that there are a lot of things that are common. We know that everything starts with their activation of the dopamine system. But then we also know that it's not identical what happens afterwards, but hopefully relatively similar. Mice you don't find in society kind of that are addicted, so you cannot do it that way. So what we have to use is behavioral models for addiction. And in principle, the results I will show you today comes from two models. One is the conditioned place preference. That basically means that you give a mouse a drug, in our case cocaine, in one compartment, and just a fake injection in the other compartment. You do this a few times, and then you open the doors here. And now the mouse will run for the cocaine paired chamber. And we see this as a kind of indicator of, first of all, that the mouse likes the drugs and that the mouse wants more of it. So therefore he moves into there. The other one is more simple in a sense that it's self-administration, or it's not. It's more conceptually simple. So the mice will press for cocaine, and you can play around with that. But that's maybe in a sense even better model than this one. One of these changes, because when coming back to this a bit, what should be emphasized here is that if you give a drug, a lot of things happens in the brain. You have hundreds of genes going up and down in a lot of places in the brain. So how can we find what is actually important? Well, that's not that easy. That has to be kind of tested. One such change that has received a lot of attention was the increase in synaptic strengthening in the dopaminergic cells. So that would be, in a sense, the core of the reward system. What we know that even after one injection of cocaine, it started with, but also all major addictive drugs, after one injection, you will get new receptors to the synapse in the dopaminergic cell. This would be a dendrite of a dopaminergic cell. This is incoming information to it. If you get more receptors here, a signal of a certain strength from this neuron will now activate the dopaminergic cell more. So it's a kind of, you make the dopaminergic cells more excitable, more easily activated. This, as I said, has been shown in response to all major addictive drugs, which makes it, of course, more interesting. It has also been shown that this depends on the NMDA receptor, which is a glutamate receptor, an ion channel. So it binds there, then this triggers some kind of event that makes this go up here. The question is here, what is the functional role of this? We cannot know. We know it happens, but we don't know how. So what we did was to take this event out by removing this one. Now, if you remove this in the entire mouse, the mouse will die. And this is not what we want to study. Dead mice we don't like, and they can definitely not be used in behavioral analysis. So there are tricks to do such things. And we used a system called the Krelox-P system. I will not go into that really now, but the effect is that you remove the NMDA receptor only in the dopaminergic cells, while it's still expressed in all other cells of the brain. Then we tried them in this test for conditioned place preference. And first, what we saw, so this would be the cocaine, this would be the saline or whatever, it's of course balanced in reality. This marks the level of a mouse that don't care, a mouse that spends equal amount of time in both chambers. So as you can see, a normal mouse will spend much more time in the place where he previously got cocaine. This mouse that has no synaptic strengthening because the lack of this receptor, he also likes the drugs and he can go there. Which came as a surprise to us actually. Now we make it go off drugs, and that's much easier with a mouse than with a patient. Because now we simply give saline here, or we just give the fake injection here, the fake injection here, for a while. And now the mouse will not care about the previously drug-paired chamber. It will just walk around and explore things. Now, we have in our model, it's of course there are potential problems with the model, but now we can induce a reinstatement or a relapse that is coming back into drug searching at least. And in this case we do this by giving a small volume of the drug and then putting the mouse here in the middle. Now a normal mouse will go and search for drug again. Here he forgot about it or didn't care anymore about this chamber, but now he's back in again. Spend a lot of time there. The mutant mouse will not do that. So here we can say that the synaptic strengthening, or at least some kind of synaptic plasticity, in the dopaminergic cells itself is important, not for the initial phases of addiction, but really for this kind of relapse-like behavior, reinstatement of the CPP. Which is kind of an important thing because it's here where you really can catch the patients, right? It's very difficult to give people pills the first time they have alcohol. It doesn't make too much sense. There are always problems with this kind of approaches. One can say that what this receptor might be important. You take it out when the animal is very young, maybe they get strange. So we use the system where we can take it out in the adult by using a compound called tamoxifen. I will not go into the details here, but the important thing is that we see exactly the same thing. Perfectly normal reward, but no relapse. These mice were also tested in the self-administration paradigm. And you could see the same there, that they learn to press perfectly. You can make them go off pressing. But if you show them something that was associated to the drug later on, they will not start to press again. Or not to the same extent, at least. So both of these studies show that synaptic plasticity through the NMDA receptor in dopaminergic cells seems to be a controller of relapse-like behavior. I will very briefly go into another thing, a related thing. So we looked at the glutamate coming in here. Next, the message goes up here. But here we know that there is also glutamate coming in. So next we studied a specific glutamate receptor that is called it's a metabotropic glutamate receptor called the mGluR5. There is a lot of this receptor here. We know from just studies where you kind of listen to the cell and look at how strong are the synapses and so on, we know that it's important for cocaine-induced changes there. So next what we did was to take out that receptor in a specific cell population up here. Here is just a picture of the cells that we target, and here are cells that we don't target. And we could show that we are kind of exact in the way we target the cells. I cut this slide a bit here, but I think you can see most of it anyhow. Here is how much these mice would press for cocaine on a lever, actually. But the wild types, so the normal mice are here in white, the mutant mice are black here. And they all press for cocaine, so this would be the basal levels. When they press for cocaine, they also see a light that they can associate to the cocaine. Here, the light and the cocaine is removed. They will press the first session, but then they understand there is no drug. I stop pressing, or I press a bit less. Mice are kind of very much into pressing anything they find, so this doesn't go back to zero at all. But and then, after here, again, the light is introduced so that when they press, they see the light, which gives them an association to the drug. And now a normal mouse will start to press again. So this is presses for light. But the mutant mice, the mouse that lacks mGluR5, will not press to the same extent at all. So this would also be a kind of relapse model. So the conclusion of that part is that mGluR5 up in the nucleus accumbens striatum is important for a relapse-like behavior in mouse. The overall conclusion is, first of all, from the beginning part, is that addiction is very complex. It's hard to study what I try to study. That would be the kind of neural correlate to addiction. That's probably quite naive thought because there are many neural correlates to addiction. It might differ a lot between people and drugs and a lot of other things. I think, however, one can distinguish at least these factors. The reward learning aspect that I talked mostly about today, the negative effect, loss of control. If we continue on the reward learning, I think the data I showed you here, and also a lot of other very recent data, mostly from animals though, I should say, is that synaptic plasticity in different parts of dopaminergic reward system are most likely very important for addiction. And drugs targeting players in these synapses might be very useful for preventing relapse in the future, which is, of course, the long-term goal of these studies. Finally, I just want to thank the labs that I've been in or collaborating with in these studies. Günter Schütz, Rainer Spanagel, David Stevens, Christian Lüscher, and also to my own lab here in Linköping. Some people working along these lines, some people not working along these lines. And thanks to you for your attention.